Hi, all. It's a great pleasure and honor to talk here for the seventh annual event in the Drug Discovery and Development Virtual Event Series organized by LabRoots. I'm Dr. Prashant Puthenbeetle. I'm an assistant professor of pharmacology in the College of Graduate Studies at Midwestern University, Downers Grove campus, Illinois. The title of my talk today is Unveiling the Therapeutic Potential of Two Sister Flavonoids Using Omics Based Approaches. So, before going into my talk, I would like to introduce my audience to some of the plant derived medicines. So, it's very important that we need to know about the importance of plant derived medicines because the topic for today's talk is about plant derived products. So the first one, as you all know, basically willow bark was used as one of the a main anti-inflammatory and analgesic agent. I mean, for long periods, and at one point, the chemists were able to make a derivative of this, which finally became the acetyl derivative of salicylic acid. So they extracted the willow tree bark, and from the tree bark, actually, acetyl salicylic acid, which in the current world we know as aspirin, was derived. Later, basically, the company which actually made the purest form of acetyl salicylic acid was in 1899 by Bayer, which is a pharmaceutical company, and they gave the name aspirin, which is chemically acetyl salicylic acid, which we all use nowadays, as an anti-inflammatory and also as for preventing platelet aggregation in cardiovascular disease. So it's like one of the widely used drug which has a plant derived origin as its own history. And the second most widely used drug is basically metformin, which is again derived from this French lilac plant, as you can see here. So for centuries, French lilac was used for many of the metabolic diseases or complications, and nobody knew exactly what was the content which was giving this metabolic protection. And only in 1920s, biguanides they isolated from French lilac, and biguanides basically is the backbone for metformin. Metformin is a biguanide, and they actually basically synthesized many biguanides, and only metformin got approved by FDA in 1995. So what I wanted to basically emphasize with this slide is basically two major plant-derived medicine, which is kind of widely used in the current medicine, especially in cardiovascular and metabolic diseases, which my lab is focusing on, is basically aspirin and metformin, owe their a contribution towards, a kind of plants. Why? Because again, they are derived from plant products. Now, having said that, that's the focus of my talk, and today's focus is mostly on flavonoids. I'm focusing on two major flavonoids. So flavonoids are, again, plant metabolites present in our food, whether it's vegetable or fruit. They're present in most of the naturally occurring vegetables and fruits, and they have, and they have basically high relevance because of their antimicrobial property, as you can see, immune defense, antioxidant property, anti-inflammatory property, free radical scavenging, hepatic protection, anti-carcinogenics against cancer, which is also this anti-mutagenic property. So because of all these properties, this flavonoids basically play a major role in human health, actually. So the first flavonoid which was isolated is saponarin, as you can see here. This is from Mosses plant, which is termed as Plagionium cuspi, datum by Kozlowski. So Dr. Kozlowski basically isolated saponarin from the Mosses plant, and that was the beginning of the flavonoid journey, as you can say. So the flavonoids are basically super family. I mean, there's like many subfamily members within the flavonoid superfamily. And the subfamily members include flavon, isoflavone, flavantriol, flavanone, anthocyanidin, and flavanol. And it's basically really tough to start, studying each and every compound belonging to the subfamily. So what I basically did was, and I'm basically trying to explain what made my interest to go towards these two sister flavonoids, or flavon subfamily members. So based on, again, the first one was literature search. So based on literature search, I was able to see many interesting properties for these two flavon subfamily members, or flavonoid superfamily members. And the first member of the family is chrysin, which has got this flavon backbone with a 5,7 hydroxy substitution. And the second product, which I'm interested in currently studying, is basically epigenin, which has also this one extra hydroxy group at the fourth position. So it's basically 4,5,7 trihydroxyflavone. And there are also other members which are like camphorol, which is tetrahydroxyflavone. That means there are 4 hydroxy group. And there's basically quercetin, which is pentahydroxyflavone, which is 5 hydroxy group. And myricetin basically has got hexa hydroxy group. So, I mean, at this point, I felt that it's very important to understand the relevance of, the prototype, which is basically chrysin, and its first derivative, I would say, or hydroxy derivative, which is naturally occurring. So it is not chemically synthesized, which is known as epigenin. So if you do a literature search, as you can see here, currently for epigenin, there are over 6,500, study reports, which include both, clinical, preclinical, and cell-based, I would say. And for chrysin, it's basically almost more than 1,800. And most of the studies basically showing all the properties which we kind of mentioned before, which is, this antioxidant, free radical scavenging, metabolically protective. So it's very important that, okay, you give this flavonoids for some indication, and, you basically highlight, or basically you target that specific indication as like, the mainstream effect of this flavonoid, whereas it may have some other, some other good and bad effects at the same time. So, what we thought was it's good to kind of restart the studies, and in a way that we understand both the beneficial properties and the bad effects of this naturally occurring agents, epigenin and chrysin. So the first step we took was, let's basically do a transcriptomics approach on these two agents and see basically what they can do to, mammalian cells. So the first one, basically, the first thing we basically thought about was, what model to start. Again, without knowing the actual dose or its bioavailability or pharmacokinetic dynamic properties thoroughly, it's not advisable to give it to a whole animal. So it's basically good to start with a cell line. So the ideal cell line we felt was mouse embryonic fibroblast, and it's very important. That's again the rationale why we picked mouse embryonic fibroblast, because they are a kind of considered primary cells, because they are not having any kind of metabolic defects or disease. They are not cancer cell lines. They are just like primary cells, but we got it from vendor. You can also basically isolate them from mouse embryos. So they are isolated during day 14 or 15 post-coitus from CD1 mouse embryos, and then the colony expanded. So it can go up to 5 passages, and up to 5 passages, they are pretty viable, maintaining the morphological and metabolic properties intact. So that's basically one of the good things about this cells, because they kind of represent the control cells. Again, like it's always a debate like which cell represent actually the whole body, and there's no definite answer for that. But at least fibroblasts are present everywhere in each and every tissue, and we are talking about a primary fibroblast from the embryo. And then again, there is a rationale behind why we picked that, because the embryonic fibroblasts are more sensitive to toxicological properties of any agents. So, if basically anything toxic is present in our, agent, that will be basically immediately be exhibited by this, embryonic fibroblasts rather than adult, which are more resistant to all kind of toxic, effects of any new agent. So again, like when we are basically doing this initial screening, we would like to know, we would like to know both the toxicological or the bad effects of a drug, and also about the positive effects of a drug. So having said that, then what we did was basically the next step, which is the transcriptomic analysis. So we simply plated this mouse embryonic fibroblasts, and we had three groups. So the control media treated group, the epigenin treated group, and chrysin treated group. So we took a dose. Again, the rationale behind the dose, like 50 is basically the maximum pharmacological dose, again, for a control cell, because beyond 50, based on reports, 60 or 75 can, decrease the viability. So we went with maximum pharmacological dose, which will then basically exhibit also the pharmacological properties of along with the pharmacological properties, the toxicological properties of the agent. And we basically incubated, our cells, which is mouse embryonic fibroblasts, with epigenin and chrysin in the media as 50 micromolar concentration for 24 hours. We pelleted the cells out, and we basically gave it for next generation sequencing as an outsourcing project with a commercial vendor. So they did the analysis. So very interesting, because we were pretty open. We were not expecting it to show any specific, activity, because we are not looking for any indication at this point. We just want to know what property these agents demonstrate. So interestingly, when we did, these are again, control cells, not any kind of disease, or they're not going through any kind of metabolic stress, just a plain control cells without meat, without serum, because we want to avoid basically the serum based effect, because serum has its own, effect on like growth signals and patterns. So we want to avoid all those confounding factors, and just wanted to see what these drugs or these agents, I would say at this point, it's not a drug. So these agents have got to do with the cells under controlled conditions. So after 24 hours, interestingly, what we saw was, so both epigenin and chrysin, demonstrated certain pathway regulation, and based on the bioinformatic analysis, based on three different pathway analysis, like wiki pathway, the reactome based analysis, and the biosec analysis, it was demonstrated that the cholesterol biosynthesis pathway was the one which was mostly regulated by epigenin. Very interestingly, when we checked chrysin, so chrysin also demonstrated a very similar effect, where cholesterol biosynthesis pathway was the one which was primarily affected, both in wiki pathway and biosec. And in reactome, a cholesterol biosynthesis came to the rank 2, but rank 1 is also SREBF, which is steroid regulatory element binding protein or factor, which is also involved in cholesterol regulation. So that's one of the interesting things. There's quite an overlap, so even though it showed a different name, the role of SREBF, which is the gene, and then the protein is SREBP, is also involved in cholesterol regulation. So unanimously, epigenin and chrysin basically demonstrated ability to regulate cholesterol biosynthesis. Now, we looked into the individual transcripts, what they're doing, and interestingly, what we found was, this is what we found. It's not like upregulating, it's downregulating most of the genes involved in the mevalonate pathway, which is the cholesterol biosynthesis pathway. And as you can see here, epigenin and chrysin, they're like both able to regulate more than over 12 target genes, as you can see. But we had to keep a screening method, because they are varying amounts of regulation. So what we did was okay, let's pick genes which are regulated over 1.5 fold. So when we set this criteria and looked into both these epigenin and chrysin regulated MEF targets or fibroblast targets, what we found was there were like 7 commonly downregulated targets in basically both epigenin and chrysin, which were involved in cholesterol biosynthesis. Very interesting. And chrysin, which is termed as compound C, has got 6 of its own, and epigenin has 2 of its own. But again, like the 7 are the one which are like over 1.5 fold decrease. So that means that these 7 targets are downregulated over 1.5 fold. Now, these are some of the major ones. I also like picked and plotted in the diagram, as you can see, and they have got a significant decrease, which along with a 1.5 fold decrease, with a significant reduction of p-value less than 0.05. So again, all this transcripts are mostly involved in cholesterol biosynthesis. Okay, so some of the commonly regulated genes in SREBP pathway, as we were trying to see, because SREBP is pathway is one of the major pathways regulated by especially chrysin, as we saw before, like in one of the bioinformatics analysis which we did based on the reactome, where the SRBF pathway, or the SREBP protein, is the one which is regulated as, which is ranked number 1 and number 3. So we were interested in finding out what are the targets which are regulated by SREBP. And as you can see here, we picked actually 6 targets in here which are commonly regulated by both epigenin and chrysin. But as you can see here, that's why it came up in chrysin more. Why? Because chrysin has almost 9 targets which are having an overlap with epigenin, and by itself it has got like 6 targets which are known to be regulated but just by chrysin itself, which belongs to both SREBP 1 and 2 pathway. But our the whole focus was how much overlap it has got with the cholesterol biosynthesis pathways. As you can see here, around 6 targets which are commonly regulated by both epigenin and chrysin were downregulated more than 1.5 fold. And in this, the IDI1, LSS, MBK, FTPS, and PMBK are the enzymes involved in cholesterol biosynthesis or mevalonate pathway. And we have one fatty acid elongation transcript, which is ELOVL6, which is also downregulated by both epigenin and chrysin. Just as a positive control, we used LDL receptor transcript, because LDL receptor is a downstream of both SREBP 1 and 2. So we basically were interested in finding out what happens to LDL receptor. And as expected, because it's a positive control for SREBP target, the LDL receptor transcript was downregulated. So along with cholesterol, so it goes back to our question that, okay, this is basically the feature shown by both epigenin and chrysin, which are like the sister flavonoids. And so the cholesterol is basically a product of acetyl-CoA. So acetyl-CoA is basically the precursor of cholesterol biosynthesis. So if you have so many targets which are downregulated in cholesterol biosynthesis pathway, where this acetyl-CoA could be going? That was our next question. So we thought like, okay, let's now really try to find out where acetyl-CoA is going. So we basically picked the rate limiting enzyme, which is involved in TCA cycle, or citric acid cycle in simple terms. And the name of the enzyme is citrate synthase, which is the rate limiting enzyme involved in the conversion of acetyl-CoA to citrate. So when we checked citrate synthase transcript, there was no change in citrate synthase. So if it's not going into cholesterol and there is no change in TCA cycle, where else it could go? So the next question is, again, fatty acid synthesis. So when we checked some of the transcripts involved in fatty acid synthesis, which include acetyl-CoA carboxylase and fatty acid synthase, as you can see, chrysin was able to downregulate both, at a significant level, which means like around 1.5-fold with a p-value, less than 0.05. Whereas Apigen was able to downregulate just fatty acid synthase, but not ACC or acetyl-CoA carboxylase as much as chrysin. But in general, in both of them, apigenin and chrysin were able to downregulate the fatty acid synthase pathway, fatty acid synthesis pathway, mostly by regulating fatty acid synthase transcript. Now, so if fatty acid synthase pathway is going down, which pathway it is promoting? That was our next question. So it's inhibiting cholesterol biosynthesis pathway. It's basically not making any change, or basically it's not regulating much the TCA cycle transcripts. So, and it's basically down regulating the fatty acid synthesis transcripts. So, or enzymes involved in fatty acid synthesis. So, where else it could be basically promoting, or otherwise, where this acetyl-CoA is actually going. So the next pathway, and one of the option could be like, okay, all the acetyl-CoA could be going into formation of ketone bodies, because acetyl-CoA forms acetoacetate, and this acetoacetate then forms hydroxybutyrate, which is a ketone body. And the enzyme which is involved in generation of this ketone body is beta hydroxybutyrate dehydrogenase, the transcript which comes as BDH1. So when we checked basically the BDH1 transcript, it was actually upregulated. So that was very interesting. And not only the transcript for the rate limiting enzyme for ketone body generation was upregulated, we also saw that the transcript for transporting ketone bodies inside and outside of the cell, which is the solute carrier 16A6, which is involved in basically, carrying the hydroxybutyrates in and out of the cell compartments, is basically upregulated. That again, almost twofold with a statistical significance. So our final conclusion based on the study one was like, just based on transcriptomic analysis, that like, under normal condition, apigenin and chrysin was able to down-regulate cholesterol synthesis, upregulate ketone body formation with no change in TCA cycle, and basically decreasing fatty acid synthesis. So having said that, our interest then became like, okay, like, what more we can know about these two, sister flavonoids. And for this, we basically use the second big approach, which is the omics-based approach, which is called metabolomics, to understand the metabolic properties of these agents. So again, the pattern is similar. We use the mouse. Sorry, I'm just getting back to the slide. So we use the metabolomics approach, and for that we basically used mouse embryonic fibroblast. And in the mouse embryonic fibroblast, again, were treated with both apigenin and chrysin. At this point, we use basically 25 micromolar, which is the sub-maximal dose. And there is a rationale behind this. Why? Because we don't want the cell to now show toxic properties, but rather we want the cell to be metabolically active and viable at the same time. So 25, is a good dose. It's a sub maximal dose. It's not like the maximum pharmacological dose, and where the cells can be viable and they can be metabolically active too. So when basically we incubated both, when we incubated MEFs with, under control conditions, and apigenin with 25 micromolar and chrysin 25 micromolar for 24 hours, and then we pelleted it out and then gave it for metabolomic study. And based on our metabolomic study, we were able to get the scattered plot, which shows the limits was set at again a log twofold change of 1.5. So anything basically, more than 1.5 fold decrease or increase would be picked by the strategy with a p-value less than 0.05. So when we set this limit, one of the things we found was the major metabolic pathway, which was regulated by apigenin, was alpha linolenic acid pathway and linoleic acid pathway. So as you can see here, this was the one which is primarily regulated by epigenin and chrysin. And this is not just based on a p-value, but also on the enrichment ratio. So, having said that, we went to look inside basically what are the metabolites actually in this alpha linolenic acid pathway, which were regulated by epigenin exclusively. And interestingly, as you can see here, what we found was icosapentaenoic acid, which is here regulated by epigenin over twofold, in comparison to chrysin, which has no effect on intracellular. So at this point, again, we are not giving fatty acid or glucose. It's just the control media without serum. The intracellular metabolic pathway has been shifted towards generation of, more icosapentaenoic acid. So that's what basically it demonstrates. Whereas the DHA or docosahexaenoic acid and docosapentaenoic acid, even though showed a trend for increase, it didn't reach the significant level. That means that in the alpha linolenic acid pathway, it was the icosapentaenoic acid which was highly upregulated at this point, at this time point. Now, when we checked what are the metabolites in linoleic acid pathway, interestingly, we found that arachidonic acid, which is mostly a precursor substrate for generation of prostaglandins and mostly, the inflammatory mediators, didn't change much. But adrenic acid, which is basically an antioxidant and vasorelaxant in property, was basically upregulated under the given condition at this time point by epigenin and not by chrysin. So exclusively, these were the properties, which was demonstrated by epigenin, which is, upregulation of some metabolites in linoleic acid pathway and also in alpha linolenic acid pathway. So we looked into what are the pathways regulated by chrysin, and we saw that in negative ion mode, chrysin upregulated alanine metabolism, and in positive ion mode, it basically regulated the urea cycle. So now the thing is how it is regulating. Is it increasing or decreasing the metabolite? The first thing we saw that in alanine metabolism, as you can see, the overall alanine formation was suppressed, as you can see here, only by chrysin, not by epigenin. So alanine in the body basically can be formed from pyruvate or by shifting equilibrium from lactate to pyruvate and then pyruvate to alanine through this GPT-1 enzyme with the involvement of glutamate and alpha ketoglutarate. So the alanine formation was basically down in chrysin treated cells, and that basically was accompanied by no change in pyruvate but basically a decrease in lactate. So that means that both lactate and alanine basically goes down in chrysin treated cells. Now the thing is, with regard to urea cycle, when we checked, and again one of the major intermediate in the urea cycle is ornithine. So when we checked ornithine, as you can see here, there is no change with chrysin compared to control, and it didn't reach statistical significance. But when we looked at some of the precursors, which is N-acetyl glutamate, which is basically comes as glutamic acid in the positive ion mode, as you can see here, was downregulated by chrysin. So the precursor for the urea cycle is downregulated with no change in the metabolites of urea cycle, which is basically ornithine, which we took as a readout. Now, glutamic acid, which is a precursor for acetyl glutamate, is also decreased or suppressed by chrysin. Now, there is a parallel pathway, basically, which occurs at the same time when this carbamyl phosphate enters ornithine. It can either enter urea cycle or it can go for orotidine pathway. And orotidine is basically the precursor for majority of this, nucleic acid, mostly pyrimidine synthesis. So, the orotidine levels were also decreased at this point following chrysin. So as you can see here, orotridine can form cytidine monophosphate, which is a pyrimidine, and it's involved in pyrimidine synthesis for nucleic acids. So cytidine and uridine monophosphate levels were decreased by chrysin, in a significant manner. So that's what we were able to see. So, along with that, some of the common metabolites which were downregulated by both apigenin and chrysin was 7-dehydrocholesterol. So as you can see here, which was downregulated by both apigenin and chrysin, and xanthosine, which is basically a precursor for uric acid. So many of the metabolites in the uric acid pathway, including xanthosine, hypoxanthine, were all downregulated, but we just put xanthosine, which was mostly suppressed by chrysin, as you can see here, like the fold increases more than 1.5. So, but definitely by apigenin too. And we just put the slide because these were the two common substrates or metabolites which were downregulated equally by both apigenin and chrysin, which had a overlapping feature when it comes to metabolite suppression. So 7-dehydrocholesterol is basically the one which is involved in cholesterol biosynthesis. So it's a penultimate metabolite of cholesterol biosynthesis, and basically xanthosine is again a precursor for uric acid synthesis. So ideally, this was our readout after the end of study 2. So the study 1 demonstrated that cholesterol biosynthesis transcripts were, or enzymes involved, the transcripts for the enzymes for cholesterol biosynthesis were downregulated by both apigenin and chrysin, and we got almost the same result using a metabolomics approach where cholesterol and uric acid biosynthesis pathway was inhibited by both apigenin and chrysin. Now chrysin by itself reduced metabolites which are involved in gluconeogenesis and inhibition of protein and pyrimidine synthesis, which is orotridine, and alanine, which is for gluconeogenesis. And that can be very helpful in diseases like both, because gluconeogenesis plays a major role in type 2 diabetes. So by downregulating, you can have beneficial effects. By downregulating purine and pyrimidines, you can have beneficial effects during cancer. Now, apigenin by itself showed, or per se, demonstrated anti-inflammatory and vasorelaxant property. That's more towards cardiovascular protective effects, along with its cholesterol and uric acid inhibitory effects. So, they have some properties which are very unique and some basically overlapping. And the overlapping ones, which are cholesterol biosynthesis inhibition, overlapped very well with the transcriptomics study, which again showed transcripts involved in cholesterol biosynthesis pathway. So, with this, basically, this was actually 2 studies. I had to go very fast. So, this was published in European Journal of Pharmacology and International Journal of Molecular Sciences for the metabolomics-based study, and the transcriptomics was the first study we did, and both from our lab, and and all the students have participated actively in the first and second, and I've been mentioned here. So so what next is basically a question. So for future direction, before making any assumption, actually, we basically need to do definitely short and long-term studies, to understand in detail, because if you just directly give these drugs for any indication, you may see some beneficial effects, but you don't know what are the other properties of the drugs. So it's very important that we understand the pharmacology of these, agents properly, along with its bioavailability and toxicological studies, both short and long term, along with drug metabolism. Once we understand this in detail, we'll be able to recommend these drugs for some specific indications. So having said that, I would like to go to the next slide, where I'd like to show the acknowledgements for a highly supportive department of pharmacology, including my chair and all the wonderful colleagues, our Dean and Associate Dean of College of Graduate Studies, and Midwestern Research Corps and Office of Research Services for all their support. The funding is, we haven't got any extramural because, again, we're struggling with natural products getting funded. So So this work was purely supported by my startup funds and the core outsourcing funds. And we had some wonderful collaborators all this while. So it's a work of 4 to 5 years. And with the help of different collaborators, including Dr. Pierre from Maryland, Dr. Kong from Loyola, Dr. Brace from KIT Germany, and Dr. Boros from Belgium, and Dr. Kurup from Ferris State. And along with that, I would like to also especially thank my students, Mr. Daniel Rafferty. They are all medical students who come for summer research program. Mr. Cole Cochran, Mr. Adan Shetty, Ms. Angela Liatnev, Caitlin, Ms. Caitlin Martin, Ms. Jennifer Choi, and I would like to thank all other students also who participated even for a short period. Was not able to put in here because of the space. So, having said that, thank you all for your valuable time. And if you guys have any question, feel free to write to me. And my email is written here. And thank you all once again for this opportunity and for your time.